Hey guys, welcome to my next episode, which is episode 4 for NSCA CSCS Chapter 1, right? So I hope you must have watched my episode 3, which was about structure of muscle fiber. If not, please watch that before actually watching this episode, right? So this episode is about muscle contraction, right? This episode is where you will learn how muscle contraction, what all things are needed for your muscle to contract, right? So starting with your muscle fiber and contraction, as we concluded, right, the smallest structure in your muscle will be your myofilaments, right? So a muscle fiber will have myofibril, and these myofibrils are long threads responsible for muscle contractions, right? So if you dig in deep, myofibrils are basically made of repeating units called as sarcomeres, right? It's like a train, right? What you see a train, right? So what you see in a train is there are multiple bogies, right? So sarcomere is like one bogie of a huge train, right? So myofibril is one train, and sarcomeres are the multiple bogies, including in that train. So what you need to understand is, again, I will break it down into step-by-step process for you to for your best understanding, right? So sarcomere can also be called as the basic unit of contraction, right? So every bogie, every train will have multiple bogies named as sarcomeres, right? So what happens when the muscle contract is basically the length of the sarcomere, right? It's like two bogies, the length of the sarcomere reduces, eventually reducing the size of the myofibril, right? So what a myofibril contains, right, will be myofilaments. These there will be two type of myofilament. One is called myosin, which is a thick filament which helps pull, right? And what will it be pulling on to is an actin, which is thin filament, which will be at the end of the I will say bogie, which is your sarcomere, right? So I'll show you the image so that you can understand it better, right? So let me just go a little further into the slide. Right, so what you see here, right? So this is a nice image that you can understand for, right? So this from this Z line to this Z line, what you see is this is one sarcomere. This is one of the bogie, right? In the center of that bogie, you will have one thick filament, and that filament will be your myosin. And the ends of the bogie, right? At both the ends, you can see this blue line here and the blue line here. That's your actin, right? So your bogie, which is sarcomere, which is contractile unit of muscle, the center is where you will find your myosin. The ends is where you will find the actins, right? So let me just see if I can uh, get maybe an arrow highlighter. Right. Right. So maybe this is your actin, right? And this is your myosin, right? And this is your sarcomere, right, which is the end, right? This is one unit where you will see muscle contraction happening, right? So what you see here is the myosin will have heads, right? You can see it here, the red ones. What happens while the muscle contract is simply the myosin heads will hold on to the actins and pull them closer from both the sides, right? So now, if your muscle to shorten, what has happened? The myosin, which has heads, hold on to the actins and pull them actins closer. So what is going to happen is basically the distance, right? This distance from Z line to Z line is going to get reduced, right? So that way the sarcomere, the bogie size is going to reduce. Now you have a train with multiple bogies. So what is going to happen is every bogie is going to get smaller, right? Eventually making this muscle to contract or shorten, right? That's what we call as a sliding filament theory, right? So what you see here is myosin thick filament. What I show you in that image was an actin, and how a muscle contract is simply pulls on to your actin with the help of head, right, attached onto myosin and pulls it, right? So what happens? The sarcomere shortens and the muscle contracts, right? So what you see here, if you look at a muscle fiber, right, under microscope, what you see here, right? So is the muscle is having something called as A band, right, which is a darker area, the area that's a little dark, right? If I can see it here, right? So what you see here, this area, this area is slightly dark because because of the thick myofilament that is myosin, right? So when you see it in a microscope, what you see is the thick filament, which is why it is a little dark in appearance. What you see light as in appearance is where you have your actins, right? So the actin, the area where the actins are, since the actin is thin filament, so what you see in microscope is that area is little light, right, compared to the myosin area. So myosin area is named as A band, which is dark, and the lighter area is named as I band, which will be mostly the actins, right? So what you see in the center here is, right, is where the myosin is attached, right? That's the center of the sarcomere or the bogie where the myosin is attached with its arms spread to the sides, right? Why the arms spread to the sides? So that it can hold on to the actin and pull them closer, right? So this place, the space from, oh, sorry, that's not, this is where the myosin is attached, right? So my bad, right? So from here to here, right? This is your Z line. This is your Z line, right? This is Z line. And this is also Z line, right? So the space, the place between the Z line is also called as sarcomere. So this is one unit. And then when you see it here, this is one unit, right? So you will have multiple units at this side, right? This is how the structure look like. So this is your thick band, which is your myosin with the heads attached. That is your actin, right? And the myosin will be attached onto the M line, right, which is here. So you'll have your myosins like this. You'll have your myosins like this. You'll have your myosins like this, right? And at the ends, you will have your actins, right? So from this, you will have your actins, which is thin. From here, you will have your myosin, which is thick, right? So myosin will have heads, right, which will hold on to the actins and pull the actin close to the body, right? So close to the center, right? So when the muscle contracts, what happens is the I band, right? The distance of the I band will and the H zone, right? So the I band and the H zone, right? I can see here. I don't see the H zone. Okay, let me not so great picture. Let me find it here. Now, see this, right? So what you see here is the distance of the I band, right? And the H zone, this one, these two, right? So when the muscle contracts, what happens here is the I band and the H zone, right? Which is this, right? So the distance and this distance becomes small, right? And what remains the same is the A band, right? What you can see here is the distance, right, of the A band remains the same because it is the dark area, right, which covers the myosin mostly, right? So when a muscle contracts, the I band in the H zone reduces or becomes narrow in size, right? Wherein the A band remains the same because that's the dark area which covers the myosin area, right? So coming back to this, what you see is ultimately is the Z line move close to each other, right? It's like the Z line is, which is again the sarcomere shortens in length, and that's how the muscle contract, right? So what you see here, this is your myosin, right? Which is, and then you have your actin, myosin heads attaching onto the actin and pulling the actin closer. That's how your muscle contracts. That theory is also called as a sliding filament theory, right? So what is important here is for you to know is for the muscle to contract, there are certain things that are needed. One is you need calcium, right? So why is calcium important? So calcium is stored in your muscle and it is stored into something called as sarcoplasmic reticulum, which you can see it here, right? The yellow ones. So for muscle to contract, you will require calcium. This calcium is essential, and this calcium is stored into your sarcoplasmic reticulum, which is here, right? And then you will have a tunnel, which is also called as T-tubules, which is again seen here, right? What you can see it here is your T-tubules, right? So basically, when the brain sends signal to the muscle, where it received? It is first received at the neuromuscular junction. From the neuromuscular junction, this impulse has to travel into your muscle, right? So there you have one chemical messenger, ACH, which I have discussed in, I think, the second episode. So this helps transmit or carry the signal from the junction into the muscle, right? And then for it to travel into the muscle, it has to travel through the T tubules, right? So these are the tunnels through which your signal is carried deep into the muscle fiber, right? So this when this happens, when a nerve signal action potential, right? So action potential is basically fastening the process, right? The process has to be really fast, because right, for me to do this, it cannot be ideally it should not take no time. This should happen within no time, right? So this process of fastening is also called as action potential. Okay, so action potential is an electrical signal that travels along a nerve or a muscle cells to carry a message, right? So which is like a fast message that's been sent from the junction into the muscle through the T tubules, right? So again, what happens as soon as the signal is received, you have your calcium released in your muscle through the sarcoplasmic reticulum, right? So now you have your calcium released into the sarcoplasmic through into your muscles through the sarcoplasmic reticulum. Okay. So what happens is calcium allows the actin and myosin to interact, right? So basically what happens, calcium is released, right? Then we need to go further here, right? So what happens basically in this is, okay, for the muscle to contract, right? So let's do this a little process, right? Let's do it this way for better understanding, right? So now, before the muscle contracts is the first phase, which is also called as the resting phase, where the calcium is low in the myofibrils, right? Most of the calcium that's there is stored in the sarcoplasmic reticulum. There is not many cross bridges between the actin and myosin. It's like the myosin head is not, I'll say very few heads are attached with the actin, right? So what happens, right? This actin, right? The actin has a binding site. It has a site where the head are going to get attached, right? These sites are not open. These sites are closed, right? These sites are blocked, and they are blocked by a, I'll say it's a chemical that's called as troponin, right? So now if this is my actin, which has a site where the head of the myosin is going to attach, but this is blocked. And this is blocked through what? This is blocked through tropomyosin, right? So there is weak interaction between myosin and actin. It's not zero interaction, right? So it's like a loose connection which cannot generate a lot of force, right? So at rest, what you see is muscle fibers are in an off state due to low availability of calcium, right? So calcium plays an extremely important role, which is why calcium low, so muscle contraction will be also low, right? So this is again to prevent muscle contraction, because you don't want your muscle to contract all the time. Otherwise, you will fatigue, right? You'll be tired. So this is the first phase. Everything is all at rest, right? Then you go to the next phase, which is excitation, contraction, coupling phase, right? What happens here, right? So what happens here is, sorry. So what happens here is when a signal is sent, right, to the muscle, basically, first thing that is happened, the signal is received at the neuromuscular junction. Through neuromuscular junction, it is transferred into the muscle fiber, right, where the calcium is released, right? From the sarcoplasmic reticulum, as soon as the calcium is released, what happens? You have your troponin and tropomyosin, right? So what happens? You have your calcium and you have your troponin. This troponin is going to go and bind with the calcium, right? Which is mostly a regulatory protein on actin, right? This is going to cause a shift in tropomyosin, right? So now this block site, and what happens? You have your troponin and tropomyosin. So your actin is potentially blocked by tropomyosin, and then you also have troponin, right? So as soon as the signal is received, calcium is released, troponin goes and binds with calcium, and this causes a shift, right? So the tropomyosin shift, and then this exposes this site, right? So once this site is exposed, what is going to happen is this head of the myosin is going to get attached, right? Is going to form a bridge, right? So myosin head is going to start attaching, right, to the actin filament initiating contraction, right? It's like getting the contraction started, right? So the amount of force, right, the amount of force that you can generate while will depend heavily on the bridge that has formed, right? It's like when I am here, I am just started with the contraction, but then when I reach here, is this is the point where I feel the strongest. I can generate max force. If I go beyond, I feel a slightly weak, right? For this, we'll have another topic, which is length tension relationship, where you'll be understanding it, where you will understand it better, right? So first stage was resting, then is excitation, the bridge formation, right? So moving further to the third phase, which is the contraction phase, right? So contraction is very important. So what happens? The myosin breaks ATP, right? So you have an enzyme called ATPase, which helps breaks the ATP. So now this ATP breaks down into ADP and phosphate. Again, it's a huge topic, so we'll not go there. I'll just concluded this with that ATP is your energy currency, right? So energy currency, whenever ATP hydrolyzes, right, energy is produced. So ATP is basically hydrolyzed to initiate something which is called a power stroke, right? So now this head has attached, but this head needs to be pulled, right? For this head to be pulled, what you need is ATP, right? So now what happens? The energy powers the myosin, right? So the ATP is hydrolyzed, powering the myosin to pull the actin towards it, right? So now both the actins pulled closer will result into shortening of the sarcomere, which is the contractile unit of a muscle fiber, right? So now what happens after a power stroke? Once the heads are pulled forward, a new ATP molecule binds with the head again, right? So what happens? See, now the heads are attached, right? So now this head has to be removed, right, for you to reach that relaxation phase. You can stay contracting tread all the time, right? So this is where the heads are pulling, right, by hydrolyzing the ATP. Now for it to detach, you also need to have your ATP molecules to bind again with the head of the myosin, right? So once this ATP molecule binds, it will help detach the head from the original position, is from the actin head, right? So if there is calcium available, right, if there is calcium, this contraction is going to happen because because the site is always going to stay exposed, right? So what happens here is, right, what is ATPase, right? So ATPase is, I say, a chemical or an enzyme that helps to hydrolyze ATP, right? What it does, ATP is the main molecule of energy, right? So ATP breaks, so ATPase, the enzyme breaks ATP into ADP and phosphate, right, which helps you produce energy, right? So that energy used for muscle contraction, simple analogy can be ATP is like a charged battery, and ATPase is the switch, right, that powers, that releases the power when needed. Okay, so ATPase is an enzyme that is found. Mama, ATP is an enzyme that's found uh, that gives energy to myosin to pull the actin, right? So, and resulting into contraction. So we go to the third phase. Now it's the fourth phase, which is a recharge phase, right? So recharge phase is where the muscle starts shortening through repeated cycles. So what happens is calcium binding continues with troponin, causing a shift of tropomyosin, resulting into the head of the myosin attaching attached getting attached to actin, right? So again, power stroke, this will this process continues as long as the calcium and ATP is available, and also the enzyme which is ATPase is active, right? So this process is also called as the, our phase is also called the recharge phase, which is important, right, depending on kind of activity that you do, right? Then you move to the last phase, which is your relaxation phase. What happens? The signal ends, the calcium is pumped back into the sarcoplasmic reticulum. Without calcium, troponin and tropomyosin return back to the original position, right? So now the binding site, right, the site where the head was attached is again blocked, and it is blocked by tropomyosin, right? So now the muscle, the head of the myosin can no longer attach, right? So this way the muscle returns back to the relaxation phase, which is also important. So simply the stepwise, if we go right, first the thing that you need for the muscle to contract is the signal from the brain, right, through a motor neuron, right? uh, so once the signal is received, then you have your release of acetylcholine, which is a chemical messenger which helps this signal at the junction to reach the muscle fibers, right? And the signal will reach the muscle fiber through the T tubules, right? So basically this is the tunnel through which the signal will move, right? And what happens as soon as the signal is received within the muscle fiber, calcium is released, right? Once the calcium is released, this calcium will bind with troponin. So this troponin will cause a shift in tropomyosin, right? So now this tropomyosin shift will allow the myosin head to fit in, right? So now this bridge starts forming. So once the head fits in, a power stroke is needed, right? So this power stroke will help the myosin to pull the actin close to each other, right? So again, for this you need ATP, and to break down that ATP, you need an enzyme that is ATPase, right? Which is already there at your myosin head. And now once the work is done, once the signal stop, what happens? Everything goes back to the uh, normal, I will say, restore back to its normal state, which is the calcium is pumped back into the sarcoplasmic reticulum. The myosin head is detached, right, and the site that was exposed for the head to be fit in has been blocked, right. So that's how the muscle contracts, right. So I hope this is a huge process that, but then all you need to know is for the muscle to contract, first thing you need is a signal from the brain. The brain will send signal at the neuromuscular junction. This neuromuscular junction will receive the signal, right? Then it has to be transferred into your muscle fiber, which is again done with the help of the chemical messenger ACH, right? And then ACH will help travel this impulse within the muscle fiber through your T tubules, right? As soon as the signal is received, calcium ion is released, the myosin head with attaches with the actin, right? ATP is hydrolyzed, resulting into power stroke, pulling the actin closer and eventually shortening the size of the sarcomere. Okay, so I hope guys you are liking my videos. I'm trying my best to make this topic really easy for you to understand and for future reference, right? So you have any uh, comment that you are, and since I record it live, so please consider my apology if I make any mistake, right? Because at times I just started with something and I goofed up, right? So I usually don't edit. This is unedited content, so please consider my apology in advance if any mistake, right? To you can always comment me for the mistake in the comment section. Thank you for your time and love. Bye.